Today we will begin one new chapter, that is biotechnology. In your CHSE syllabus as well as the CBSE syllabus, this biotechnology includes two chapters. The first chapter is principles and processes of biotechnology. The second chapter is applications in biotechnology. So first starts with principles and processes. So before that, we want to know what is biotechnology. You can see the word biotechnology includes two things: biology plus technology. Biotechnology, that means technology means scientific application of scientific knowledge so that we can produce products for human welfare. Scientific knowledge को utilize करिकी जदि आमे किसी नया प्रोडक्ट तैयारी करिवा, that is technology. So what is the role of biology in technology? So biotechnology includes the techniques of using organisms or enzymes of the organisms to produce products and processes useful in humans. So from this we can say that biotechnology is nothing but production of curd, bread, wine. That is traditional biotechnology. In our day-to-day activities in our house, every day we prepare curd. In the laboratory we can prepare wine, alcoholic beverage, but that is not enough to supply to entire population. So it needs industrial scale production. So that industrial scale production involves the techniques of biotechnology. So biotechnology has two meanings: traditional biotechnology, modern biotechnology. In traditional biotechnology, curd, bread, wine making process involves all microorganism mediated processes. But modern biotechnology involves other techniques also. For example, test tube baby formation, then synthesizing gene, that means artificial gene construction, and using that gene for production of various gene products. For example, insulin. When beta cell of pancreas stops secreting insulin, that means the individual is affected with type 2 diabetes. Diabetes 2 type, type 1, type 2. So when diabetes mellitus occurs, that person needs injection, insulin injection. So that insulin can be produced in industrial scale by means of synthesizing artificial gene. We will discuss in application portion of the chapter in the next few days. Then preparation of DNA vaccines, then correcting a defective gene, then various other techniques, production of pharmaceuticals. That is also the contribution of modern biotechnology. Today we can test everything in a rapid manner, that is molecular diagnosis. All are because of modern biotechnology. So far we discussed what is biotechnology. EFB, European Federation of Biotechnology. This organization defined biotechnology, which includes both traditional view as well as modern view. What this EFB say? According to EFB, biotechnology is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. So, this is the definition of EFB regarding biotechnology. Now, biotechnology includes these two core techniques. One is genetic engineering, otherwise called recombinant DNA technology, RDT. So, this technology involves the construction of recombinant DNA. That's why genetic engineering also known as recombinant DNA technology. This is the first core technique. Then second one is chemical engineering. So we'll discuss now what are these two things. Engineering means manipulation. Manipulation, what type of manipulation? Here we are going to manipulate a gene. From molecular biology, you know gene is nothing but a DNA segment. DNA is a very long molecule. It is the genetic material. So, one segment of DNA that can express in two-step transcription, translation. So, suppose this DNA is mutated. So, this mutated gene is not functional. So, we need another functional gene which can replace this defective gene. So that is engineering. Suppose a student obtain 8 marks in his exam. So he or she can manipulate his mark just to show his parent or her parent by just placing 1 0, and it is now manipulated to 88. Pen is for writing. I can manipulate the use of pen for scratching my back, creating sneezing stimulus, or you can throw to your friends sitting before you. So all these things are not the real use of pen. You are just engineering the pen as per your need. Pen is required for writing, you are engineering for other purposes. Similarly, genetic engineering involves the manipulation of DNA or RNA. Manipulation of DNA or RNA. That means we can change the chemistry of DNA or RNA. After that, what we should do? We should introduce into host organisms. Then what should we do? So that we can change the phenotype of the organism. So in genetic engineering, basically 3 things taking place. First, we are changing the chemistry of a gene or DNA. Then we are introducing that changed chemistry of gene into a cell or organism. Then we can culture it. So here the initial definition giving you the idea. In genetic engineering, the chemistry of DNA or RNA is changed, which will be introduced into host organism so that phenotype of the organism will be changed. Suppose this is a gene introduced into a cell. So earlier this cell is not having this gene. So this is a foreign gene, foreign DNA for this cell, or we can say alien DNA. A-L-I-E-N, alien means foreign. So when the foreign gene is introduced into a cell, this cell is now called a transformed cell. Transformed. So transformed cell means the cell is having a foreign DNA, or I can say it is genetically engineered cell, or I can say it is a genetically modified cell. I have modified its genetic material by just introducing a foreign DNA into it. So, genetic engineering basically involves three major steps. This tick mark. So, what are the steps of genetic engineering? Genetic engineering or recombinant DNA technology involves three major events. First, creation of recombinant DNA or construction of recombinant DNA. How we can construct it? These are the sub-steps. Leave it now. Then second, gene transfer. Recombinant DNA includes two things. One is our gene of interest plus another is vector DNA. So when two different sources of DNA united, then it is a recombinant DNA. दोनों जीन DNA का recombination हुआ है, reunion हुआ है. So when the DNA from two different sources ligated, united, linked, joined, that is the recombinant DNA. So once recombinant DNA is constructed, that DNA contains the gene of interest, this foreign gene. So introduction of recombinant DNA into a cell is gene transfer. Or I can say the process is called transformation. The cell is called a transformed cell, or I can say it genetically modified. In definition of genetic engineering, I told you, genetic engineering involves changing the chemistry of the DNA. This DNA alone cannot undergo replication. It cannot increase its number. That's why we are joining into it another DNA, that is vector DNA. So in recombinant DNA, two types of DNA present, that is gene of interest plus vector DNA. So the gene of DNA, gene of interest can be called as a passenger DNA. My motorbike is a vector vehicle. So this is a vehicle DNA, vector DNA. So this DNA is carrying my gene of interest. You can ask why we should not directly introduce the gene into a cell. Why we need vector DNA? For your information, what is the structure of a gene? Recall from the molecular biology chapter. Molecular biology ro मन रखा structural gene ro आरंभ रे beginning has a promoter and here is the terminator. One is template strand, polarity 3' to 5'. The polarity 5' to 3' is coding strand. Only one strand will express, and this DNA, when introduced into a cell, this DNA cannot undergo replication. From molecular biology, you know, replication begins from a site called origin of replication or oricite. So this gene is not having any oricite. That is the problem. So this oricite is absent in the gene. That's why the gene alone cannot replicate to increase its number. When a molecule increases its number to produce identical molecule, that is cloning. So unless oricite is not there, this cannot replicate or cloning not possible. That's why the vector DNA. Now it is clear. So in construction of recombinant DNA, we need two type of DNA. One is gene of interest and another is vector DNA. So what is the purpose of vector DNA? Vector DNA contain oricite. That's why the vector DNA can replicate, and along with it our gene also will replicate. So that's why this passenger DNA, the gene, should be joined, should be linked with the vector DNA. So this joining involves one enzyme. When we are joining to paper, we are using the gum, GUM. Here, these two molecules need some gum. That is nothing but enzyme. That is called molecular gum ligase. Ligase. Ligase enzyme involves the joining of these two different DNA. Now, creation or construction of recombinant DNA, that recombinant DNA contains gene. So gene transfer. Gene can be transferred into a cell. The process is transformation. The cell produced is transformed cell or genetically modified cell or genetically engineered cell. So this genetically engineered cell should express the gene. Suppose this is insulin gene. Our purpose is to prepare recombinant or genetically engineered insulin so that it can be given to entire population, to the entire world. So for industrial scale production, this recombinant cell should multiply to increase its number. So every cell will produce insulin. So my quantity of insulin, volume of insulin will rise. So there comes a point, gene cloning. So gene cloning involves the technique so that the DNA multiply its number so that identical DNA can be obtained. That is the cloning. So this is the summary: creation of recombinant DNA, gene transfer, gene cloning. Here in your NCERT book, this is the summarized diagram representing recombinant DNA technology. You see here, this is the very long DNA, chromosomal DNA. Suppose this is a DNA from our chromosome. This chromosome contains insulin gene. So after isolation of DNA, what we have done? We fragmented the DNA into segments. This also involves some enzymes, that is restriction endonuclease. From this fragment, we selected the DNA fragment that is a gene of interest. I told you this gene alone cannot replicate it because it doesn't have any polycyte. That's why we have to select a one vector DNA or vehicle DNA. So that is a plasmid. So what is a plasmid? Plasmid is a extra chromosomal circular DNA. Where it is present? Suppose this is a bacterial cell, this is the nucleus, and these are the small ring-like DNA molecules. So bacterial cell ଭିତରେ nuclear DNA is the main DNA. This small ring-like DNA are plasmid DNA or vector DNA. So these vector DNA should be isolated. Then we will cut the both gene as well as vector DNA so that we can obtain similar type of end. When their end will not be similar, they will not fuse, they will not join. That's why we are using the same enzyme. The same restriction enzyme will cut here, the same enzyme will also cut the vector, so that their end will be similar, their end will be cohesive, so that they can easily fuse. So this is the construction of recombinant DNA. So this recombinant DNA will be introduced into a cell. So once it is introduced, I told you the process is transformation. The cell is a transformed cell or genetically engineered cell. So this genetically engineered cell undergo cloning. So 3 steps, you got it. Now you see what happens after cloning. So this cloning process can be done in a huge industrial scale inside a bioreactor or fermenter. You know it from microbes in human welfare. So how alcoholic beverages prepared, how dairy products prepared. Huge stainless steel container. In that container, 2 things present: nutrient medium and the genetically engineered cells. That's why chemical engineering. So chemical engineering means we will engineer the nutrient medium. That nutrient medium should contain antibiotics so that other cells should not interfere. ताले chemical engineering रे कौन करा हो? गोटे sterile medium create करा हो. से sterile medium टा ए genetically engineered cell को culture करियारे साहाय्य करिवो. This genetically engineered cell requires some extra nutrients. So we will prepare the culture medium which will contain this extra nutrients for cultivation or culturing. After that, downstream processing. Downstream processing means this is the method by which we will separate and purify our product. All of you know the gene of interest will express inside the cell, so it will become the product. The product of recombinant DNA technology. So we need the isolation of that product, purification of that product. These two things together called downstream processing. So downstream processing involves separation and purification. Then quality control testing. So every food products retain its manufacture date. You can see one medicine bottle. There you can see batch number. That means thousand bottles prepared in the same batch, all are having same number, all derived from the same fermenter, so same batch number. So quality control inspector, one technician is there who will test its quality as per the international standard. It is suitable for marketing or not. After quality found good, certificate will be issued, register will be maintained. Suppose say medicine टा खाई कौन लोकर अनिष्ट घटला ताले सही बैच नंबर को इन्स्पेक्टर जांच करथिला केबे कार सेटा डुप्लीकेट मेडिसिन ना नहीं inquiry हो. That's why quality control testing also necessary. Then finally we will send the genetically engineered product, recombinant protein, to the market. All these steps together one long question for CHS. In this long question you have to discuss each and every side with appropriate diagram. Clear? So biotechnology involves two core techniques: genetic engineering and chemical engineering. In genetic engineering we manipulate the chemistry of DNA and then introduce that DNA into the cell, and then we clone it so that the phenotype will be changed. Then now we will discuss creation of recombinant DNA one by one. Before that, I should say the tools of recombinant DNA technology. Then we will discuss the techniques. Tools means instruments. So in our motorbike, that is a tool kit. Means that box contains various instruments. If anything happens during our long drive, we can do some repairing work. That's why the tool kit. Here engineers tools, genetic engineers tools involves various enzymes. This is one of the important tools. What are those enzymes? First one is lysing enzymes. Lysis means to break down, to dissolve, and all these lysing enzymes to degrade the cell wall and the cell. आमे कोटो डीएनए पाइबा सेल भितर पाइबा ताले सेल टाको बांधिया को पड़िबो ताले से ब्रेक डाउन करिया पई लाइसिंग एंजाइम दरकार. We'll discuss it later. Then डीएनए पाइला परे डीएनए टाको फ्रैगमेंटेशन करियो ता पई को एंजाइम दरकार. रेस्ट्रिक्शन एंडोन्यूक्लिएज दरकार. It is very important. 2 marks short note for your exam point of view, 2.5 marks. Then after the DNA fragments obtained, we will fuse it with vector DNA, two different DNA, so it is DNA ligase. So this is called molecular gum. अट्ठा भलिया काम करछि. This is called molecular scissor. ये काटिबो DNA इंटरनल साइड रे कैंची भलिया काम करे. Molecular scissor. Then TAC polymerase, TAQ. It is the short form of Thermus aquaticus, one archaebacteria. So TAC polymerase. Why it is necessary? It is necessary, this enzyme is necessary for amplification of desired DNA. All of you know inside the cell, DNA replicate because of DNA polymerase. So this is a DNA polymerase, TAC polymerase. Why it is called TAC? Because it obtained from a archaebacteria name. Its genus is Thermus aquaticus species. So this archaebacteria contain the enzyme TAC polymerase. So this is helpful for increasing the number of DNA in a test tube. Test tube भितरे indefinitely कौनसी जीन रो संख्या को बढ़ाया पाई आमे जो टेक्निक फॉलो करिवा that is PCR technique, polymerase chain reaction. से टेक्निक रे ए एंजाइम रो यूज हो. And another one is alkaline phosphatase. Alkaline phosphatase. ए एंजाइम का फॉस्फेट टर्मिनल फॉस्फेट ग्रुप को भांगिबो. तो को मॉलिक्यूलर बायोलॉजी रे पढ़ा हैछला. DNA रो 5 प्राइम एंड 3 प्राइम एंड तमे बॉय रे डायग्राम रे देखिबो. एनसीईआरटी बुक रे एठी देखो 5 प्राइम एंड रे फॉस्फेट अछि. एमती जदी दीटा DNA फ्रैगमेंट जदी जोडी गले यार फॉस्फेट 5 प्राइम तार 3 प्राइम तो दीटा ऑटोमेटिकली लाइगेशन हो जिवो. से तम परमिशन मागिबेनी. आमे तो सुडा को चाहनो. तेणु आम वी शुड एवॉइड दिस टाइप ऑफ ऑटो फ्यूजन. ताले आमको कौन करिया पड़िबो ना या फॉस्फेट को कट कर दो पड़िबो. ताले अल्कालाइन फॉस्फेटेज देकी जेतेबेले मो टर्मिनल फॉस्फेट ग्रुप को रिमूव कर देबी ताले ए डीएनए टा का संगे लाइगेशन नहीं परिबोनी. ताले ऑटो जॉइनिंग कैन बी प्रिवेंटेड by using this enzyme. बुझेला. Now various type of enzyme छोड़ा आमे कौन दरकार ना रिकॉम्बिनेंट डीएनए तैयारी करे जो सेल भितरे मो ट्रांसफर करिबी गोटे होस्ट सेल दरकार ना. तेणु से होस्ट सेल शुड बी कॉम्पिटेंट. ब्रैकेट रे मो कॉम्पिटेंट लिखि होछि. कॉम्पिटेंट होस्ट सेल तमाने से एटा भी गोटे टूल्स. तले मो आर्टिफिशियली रिकॉम्बिनेंट डीएनए कंस्ट्रक्शन कला परे आई नीड टू इंट्रोड्यूस दैट रिकॉम्बिनेंट डीएनए सुइटेबल होस्ट सेल एंड दैट होस्ट सेल मस्ट बी कॉम्पिटेंट इनफ. जदि गोटे बैक्टीरिया सेल कॉम्पिटेंट नाही मु तार भितरे गोटे आरडीएनए भर्ती कर देछि दैट बैक्टीरिया विल डाई ड्यू टू शॉक. स्टूडेंट को काली आज जदि न्यूज रे देखि दिया हो 5 दिन परे काउंसिल परीक्षा स्टार्ट. तो तुम माने तो प्रिपेयर्ड नो. दैट मींस यू आर नॉट कॉम्पिटेंट टू हैंडल दिस कैलामिटी. तेणु तुम कौन कर आउट ऑफ शॉक हार्ट अटैक हो. बिफोर इंट्रोडक्शन ए सेल को कॉम्पिटेंट करने को पड़ो. किसी केमिकल दरकार टूल्स अंडर आसिला. देन सुइटेबल कल्चर मीडियम जोटा केमिकल इंजीनियरिंग रे आसुची ए होस्ट सेल ता भितरे रिकॉम्बिनेंट डीएनए इंट्रोड्यूस कला परे से होस्ट सेल ता को कोन कहबी जेनेटिकली इंजीनियर बा ट्रांसफॉर्म सेल से ट्रांसपोर्ट सेल ता को खाद्य देबे ना नहीं ताले आर्टिफिशियल किछि फूड मटेरियल प्रिपेयर करिवि दैट इज द पार्ट ऑफ केमिकल इंजीनियरिंग तेणु से केमिकल तैयारी कला परे भाग माप करके देबी तेणु सेल गुडा क्लोनिंग हबो ताले सेथरे पड़े आसिला सुइटेबल कल्चर मीडियम एटा भी टूल्स रे आसिला सुइटेबल कल्चर मीडियम so these are the basic tools of genetic engineering then first we will discuss how we can isolate DNA. No, let's begin with this enzyme first. Today time is short. Restriction enzyme, endonuclease. So all of you know, nuclease is the nucleic acid digesting enzyme. Nuclease is the nucleic acid digesting enzyme. So, restriction enzymes. Nuclease can be exonuclease, endonuclease. That means, suppose this is a DNA molecule. When this DNA molecule will be provided with nuclease, it can degrade from outside. Exo माने बाहर degrade करी जिवो. Endonuclease can break the DNA in the internal site. भितरे भितरे भांगी परिबो. तो एटा को हम कहले endonuclease. Coming class discussion करिए.